Hello, good morning everyone. Today we are going to start with our fourth unit, that is the muscle physiology. We've done the general physiology, cell physiology with the membranes. Now physiology also we completed in the last couple of days. We will follow the same pattern for the muscle physiology also. So there are more than one type of muscle in our body. What are the muscles of the body? The classifications like the muscles of the body are the skeletal muscle, we have the smooth muscle, and our cardiac muscle. Today we are going to discuss the skeletal muscle. Now, what are the salient to start with? What are the salient features or general features? Salient features of a skeletal muscle fiber. Okay, what are the general features of a skeletal muscle fiber? Location, it origins, that it begins, and ends, that is inserts in a tendon. Okay, so I'm drawing it like this. Say this is the bone and this is the tendon. So how, in what hierarchy does this skeletal muscle stay? It is something like this, and this is, say, a cross section. So this outermost covering, which is a connective tissue, obviously tough connective tissue, it is the epimysium. Sethi Usman, good morning, nice to see you again. Then it holds a group of the fascicles. Okay, like this. These are the group of the fascicle. Now, each fascicle is again enclosed by another connective tissue covering, which is the perimysium. Now, each fascicle encloses this muscle fiber, like single muscle fiber, which is covered by another connective tissue, which is endomysium. And this is, if we draw it, this is the tendon, and this is the bone to which the muscle through the tendon is attached. So this is the hierarchy. Now, if I highlight one muscle fiber, it will stand out to be like this, like this. Now, each single muscle fiber of the skeletal muscle, and also cardiac muscle will come later, skeletal muscle, each single muscle fiber is termed as myofibrils. So what does it stand as? Like each myofibril is enclosed by a connective tissue which is the endomysium. So many myofibril together forms a bunch which is enclosed by perimysium, another connective tissue, and that forms a fascicle. Many fascicle together is enclosed by a outer tough covering which is the epimysium. Now all this perimysium, epimysium and endomysium, they are connective tissues, and these connective layer is made of collagen and a protein, that is elastin. Okay, this is what they are made of, collagen and elastin. Okay, other salient feature like characteristics feature of a skeletal muscle fiber is that this is like the first one. The second characteristics feature is like they are multinucleated, okay, striated, and has organelles like mitochondria. So what is a mitochondria in a skeletal muscle called? All of us know that it is called as sarcomere. Then what else? What are the common organelles it has? It has got smooth endoplasmic reticulum, which is called as sarcoplasmic reticulum, right? And it is also having Golgi apparatus. Now, the cytoplasm of a skeletal muscle fiber has glycogen. Okay, skeletal muscle fiber is also rich in a pigment called myoglobin. These are the general features or salient feature or structural features of a skeletal muscle fiber, and they look striated. They look striated under microscope. They look like there are striations. So it is something like this. Okay, it's like multi-nucleated, and there are like striations also. Okay, roughly it will look something like this in it under light microscope. Okay, coming to the function. What is the function of the skeletal muscle in our body? Function is mainly maintenance of posture. Like when we are sitting, when we are standing, when we are walking, we are indulged in so many activities, right? Physical activity from morning till night. So maintaining the posture is one of the essential function that the skeletal muscle carries out. This is the first function. Second function, it aids in motor function. By the contraction and relaxation of the skeletal muscle, we can do the movement. Like when we are sitting, when we are standing, we have to walk, we have to run, we have to crawl, we have to stroll. Everything that we are doing, we are aided by the skeletal muscle. Apart from that, this, it stores nutrients and also oxygen. We know that how, because of the myoglobin, it stores nutrients and oxygen. Then it helps in maintaining body temperature. Okay, these are the functions of a skeletal muscle. So skeletal muscle means its unit is the myofibril skeletal muscle fiber. So this, all these myofibrils, the skeletal muscle fiber together, they are helping in carrying out these functions. Okay, coming to the important structure, that is the sarcomere. That is the sarcomere. By definition, sarcomere is, it is the contractile unit of skeletal muscle. Okay, it is a contractile unit of skeletal muscle. Now, if we study a muscle fiber, that is a myofibril, under a light microscope, it will look something like this, in which some portions are darkly stained. It will appear like some portion, it has got this dark area, I've got this dark areas. Okay, and some are having lighter areas. Some are having the lighter areas. This lighter area and darker area, it is happening. Why? Because of the refractive index, following the principle of physics, that and all we don't have to bug our heads with. What we have to remember is that the darker area, this is called the A region, A region, A region, A region, and the lighter areas are called the I region, I region. Okay, here this A region or band is anisotropic. Okay, anisotropic. Whereas the I region or band is isotropic. Okay, anisotropic means where the refraction of the light is less, and isotropic means where the refraction of the light is more. Now, if we take this area, and we enlarge it. Okay, if we take this area and enlarge it, so I'm going to draw an arrow that will take us all the way till here. Okay. Now, this is also under the light microscopy. How does this region, if I expand it, how will it look? It will look something like this. Okay. In between, we have the A band. On either side, we have the I band. Like one, I'm just taking one region, say this one on the either side of it. Okay. So this A band is the thicker one, I band is the not so thick one. Now again, from the middle of this A band, there are thicker line that is kind of little bit bulging out is noticed, which is the M line. Now, in this thick region itself, this M line is like little bit more thick. It's like little bit more thick. This M line. Okay, just enlarging it little bit for our better understanding. Sorry. Okay. Okay, this is the M line. Sorry. Now, from the either side of this I band, a line penetrates all the way between the I band. Now, this is the Z line. This is the Z line. Sarcomere is the area between the two Z line. Okay. Sarcomere is the area between the two Z line. And the not so bright area on the other side of the M line, it is the H band. It is the H band or Heller band. Now, this is what we see under microscope, light microscope. Now, what actually makes this A band thicker and the I band thicker? Now, we know that a skeletal muscle fiber is having the contractile protein, actin, and myosin, and sarcomere being the contractile unit obviously have this too. So again, if we draw the A band and I band, it will stand out to be like this. This is, say, the A band, which is having the thick filament, which is the myosin. Okay. Now, on the either side are flanking what? On the either side, the flanking structures are the actin. Okay, are the actin. Okay, so this is the A band, the thick one, and this is the I band. This is the I band. And this structure which is right in the middle, passing through the I band, is the Z line, is the Z line. So here you see why A band is thicker than the I band, because here you see there is a region where there is overlapping of the actin and myosin. So this is the actin and these are the myosin. So this area of the A band, here there is overlap of this actin and myosin, and myosin especially, it is like thick filament. That's why the refractive index is dark. It is dark, and here runs the M line. But again, on either side of the M line, on the either side of the M line, what do we see? We see there is relatively little lesser dark region. It is slightly lesser dark region. So this slightly lesser darker region is the H band or the Heller band. This darker region. Okay. In a sarcomere, it holds, like in 1 unit of sarcomere, it holds one A band plus half I band on either side. So you see here, this is like Z line to Z line in a sarcomere. Just if you look at the picture, you will understand what I mean to say. This whole thing is the sarcomere. So Z line to Z line. So Z line is having, see here, half of the I band, half of the I band on the right side and the left side, and it has one A band. And this is in the resting condition. This structure is in the resting condition. In contractile state, the A band and I band get closer. So how does it look? How do they look actually? They look like this. In contractile state, they look like this. So this is the thick filament, the myosin with the M line in between, and during contractile state, the actin and the myosin overlap is more. The actin is much more closer to the myosin. Okay, it is much more closer to the myosin. So here, if we compare the picture to this, what do we see? We see that the A band here and on either side, this is the I band and I band. So during contraction, the A band remains constant, whereas the I band moves closer and reduces in length, right? This is what is happening. See, this is our M line. These are the Z line and Z line. So see, compared to this picture, if you see this one, the length of the A band is same. It is not changing. What is happening? The I band is getting pulled inside. The I band is coming much closer to the A band. That is the reason why the I band length is getting reduced. Okay. So that means the contractile unit sarcomere is doing its physiological function. Okay. This is our sarcomere. Coming to the proteins of the muscle. The muscle is rich in protein. It has got so many protein for its both structural property, structural purpose, as well as the functional property. The four main types of protein that a skeletal muscle fiber is having are number one are the contractile proteins. Okay, the proteins that carry out contraction of the muscle. Second, we have is the anchoring or stabilizing proteins. Okay. Third, we have the regulatory proteins. Okay. And fourth, we have the relaxation proteins. These are the four type of proteins that is present in a skeletal muscle. We go into them one by one. Okay. So, first, contractile proteins. Okay. What are the contractile proteins? So, contractile proteins, they are the actin and myosin. Starting with actin. What are actins? These are, first, there are monomers of actin protein called as sorry, G actins. G actins. So they are something like this. Okay, they are the monomers. Okay, this is a G actin. Now, G actins form diamonds like this. Like this. Okay, they form dimer. Okay. Now, this dimers then polymerizes to F actin. So now polymers of F actin are formed, and this F actin forms a helical structure. Okay. So this is a helical structure of a F actin. So this is what is a polymerized actin protein which is contractile in nature. Second is myosin. Second contractile protein is myosin. And one more thing, this actin, it is also called as thin filament. Okay. Now, myosin also called as thick filament. It has also got a specialized structure. It has subunits 1 to 6. Now, in skeletal muscle, myosin 2 is present. Now, what is the structure of this myosin? The structure of this myosin is something, structure in the parts of myosin, it's something like this. Okay. Now, this is like called the heavy chain. This tail portion, this is the tail portion, and this is made of heavy chain, and it is helical in structure. Now, here lies a comparatively, like it lies a comparatively flexible region, which is called the flexible neck region. Now, myosin molecule, it has got two globular head. It has got like two globular heads. Now, this globular head, they have they have light chains. They have, these are the MLC, myosin light chain, which is activated during the process of contraction. Now, this has got binding site. Okay, this myosin light chain, they play a very crucial and important physiological role. Okay, they play a very crucial and important physiological role. So, one side, it is the ATP binding or catalytic site. Another side, it is the regulatory site, also known as the myosin binding site. So, this is how a typical myosin molecule or myosin protein looks. So, both are the contractile protein. Moving on to the anchoring or structural proteins. Now, what are the anchoring and the structural proteins? Which are they are also of more than one type. Okay, we have the cap, Z, and actinin. Now, you say this anchoring and structural proteins, they are, by the name itself, we can understand that their main function is to anchor, is to maintain the structural integrity of the skeletal muscle fiber, because it is contracting, it is relaxing. So, in doing so, it should not become like loose and come out, or become flaccid, or lose its tightness. All these things should not happen. It should not get cut out from the tendon also. So, this protein actually maintains the elasticity, and it hooks also this skeletal muscle to the tendons. So they work all across the skeletal muscle fiber so that it can maintain its mechanical and structural stability. So capsi and actinin protein, what they do, they connect the actin to the Z line. Okay, the Z line and the actin are connected by this capsid and actinin protein. Second, we have the titin, which is also known as the connectin. Now, this is the largest protein of the muscle. And what it does, it connects the M line to the Z line. Okay. Next is desmin. So desmin, it is the intermediate protein. What it does, it connects the Z line to the plasma membrane. So during the shortening of the I band, during contraction, it does not tear away, it does not get torn or distorted because of this elastic proteins, the titin, the desmin, they keep them integrated. Then we have another protein which is nebulin. Now, it maintains the elasticity of the skeletal muscle fiber. It runs through the skeletal muscle fiber and it maintains the elasticity of it, especially helpful during the contraction and relaxation. It has the myomycin. Now, this protein is present in the M line. So whenever there is contraction, there is a little bit of bulging of the M line towards the front, but it does not snaps away. It does not get torn away. Okay, so this is maintained by the myomycin. We have the DGC or dystrophin glycoprotein complex. What does this do? This protein maintains the stability and helps in scaffold formation. Okay. Now, examples of these proteins are dystroglycan, merosin. Okay. And sarcoglycan. These are the important proteins that are helping in the anchoring process. Third, we come to the regulatory proteins. Now, what are the regulatory proteins? The regulatory proteins are tropomyosin and troponin. Now, both this protein, what is their main function? These proteins inhibit, this protein inhibits the actin myosin interaction during relaxed state or until and unless the contraction is needed. Okay. Drawing the tropomyosin. Okay. Now this is the tropomyosin. What it does, it enters through the grooves. It is like grooving. It is grooving between this actin filament. This actin is lying as a double helical. Okay. It is grooving across. Okay. In between, it is like attached to the actin and it is like grooving. Okay. This tropomyosin is inhibits the actin myosin interaction. Now, the second regulatory protein, which is the troponin. Drawing a segment of this tropomyosin along with this actin, it will stand out to be like this. Say, this is the tropomyosin and these are the actin filament, just a segment of it. This is actin, this is tropomyosin. Here also, these are the actin. Okay, so troponin is lying something like in this orientation. Okay, something like in this orientation. Now, this is our troponin. Troponin has three subunits. What are the three subunits? Troponin T, troponin I and troponin C. Okay. Now, through this troponin T, through this troponin and tropomyosin is attached. Okay, that is through the subunity, the troponin and tropomyosin are attached. Through this troponin I, the binding site of myosin on actin is covered. It covers, it like flaps over the site on which the myosin head will bind. Okay. Whereas troponin C, it is the calcium binding site. So these are the subunits of the troponin with its function. So whenever there is no contraction needed, this troponin I will cover the binding site on the actin on which the myosin will bind. And C is the one in which the calcium will bind, because for any contraction to happen and relaxation to happen, calcium is needed, which we will study in our next class. Okay. Remained is the relaxation protein. Lastly, now what are the relaxation protein? Okay. To ensure relaxation and not too much of contraction, not only the termination or the physical contact of the actin and myosin has to be removed. Okay. The quantity of the calcium that has come to the cytoplasm, that is sarcoplasm, has also to be thrown out of the sarcoplasm. So relaxation protein, their main function is to remove relaxation protein. Their main function is to remove the excess calcium from the sarcoplasm. We know what is sarcoplasm. It is the cytoplasm of the muscle cell. So to remove the excess calcium from the sarcoplasm. Now, two proteins are very active and important in this. One is the Sarca pump or Sarca protein. Another is the calciquestrine. And remember, all this muscle protein that we are discussing now is in skeletal muscle, fiber. Okay. So these are the two proteins. Now, what is the function of this protein separately, individually? Now, Sarca pump, it removes the excess calcium after contraction back into the sarcoplasmic reticulum. So when there is no calcium, there is no trigger for the binding of actin and myosin to happen. Okay. Second is the calciquestrine. Now, how does this calciquestrine act? This calciquestrine, what it does is it buffers. It does not remove. It buffers the calcium remaining in the sarcoplasm after contraction. Okay, so these two are the muscle proteins. Okay, so we have studied the structure and function of a skeletal muscle. We have understood the sarcomere and its function and its structure totally in details. We have also studied the electron microscopic structure of myosin and actin, discussed its various parts, and also the role of the various protein that is present and are important in carrying out the function of the skeletal muscle. Okay, moving on to a new segment, that is the uh, special type of a synapse. It is a very special type of a synapse which is called as neuromuscular junction. Now, this synapse is not only very interesting in structure, but physiologically, functionally, clinically, pharmacologically, it is also a very happening area, a very happening place where lots of mechanism happen, and it is also a site for various drugs to act on. We have to understand this neuromuscular junction in total details, So, going by the definition. What is a neuromuscular junction? This is a meeting point which we call as a synapse. In medical term, it is synapse, right? Scientific terminology. It is a meeting point between an alpha motor neuron from the spinal cord, obviously, and a skeletal muscle fiber. Okay, so this is a junction or a synapse or a meeting point between the spinal alpha motor neuron and a skeletal muscle fiber. Okay, this is what is a neuromuscular junction, and this is a very special junction. It is just not any type of a junction. This is a very special type of a junction in which many activities happens. Okay, so what is happening in this junction? Okay, let me keep the chat open. Okay, it's open now. So anyone, if you have any doubt, just feel free to interact through the chat. So, going by the structure, light microscopic structure, how does it look under light microscopic structure? Okay, so here it looks something like this. Okay, where the axon, the terminal part of the axon, has lost its myelin sheath, and through the foot process, it is innervating a folded like structure of the skeletal muscle fiber. So this is like this is the axon. Okay, axon of what? Axon of a spinal motor neuron which lost its myelin sheath, and it has like gone and supplied to a skeletal muscle fiber whose membrane is folded into a structure which is called as a palisade. Now this area is called the end plate. End plate is the structural thing we are talking about. Okay, end plate or motor plate, both mean the same. Now here, if we look into a bigger electron microscopic structure, the structures are much more defined and much more prominent. Okay, before moving on to that, just remember, whenever there is an action potential generated and transmitted through the axon, it releases acetyl-CoA. Now, acetyl-CoA is excitatory neurotransmitter. It is seen that even in normal condition, there is always a few molecules of acetyl-CoA in the motor end plate, which is depolarizing in nature. Okay, so there is always little action potential in the end plate known as the motor end plate potential. So what happens is that there are this sodium channel, sodium channels. So their influx through them passes the wave of depolarization. Now, when the depolarization wave passes, it induces the release of this acetyl-CoA. Acetyl-CoA. Okay. So this acetyl-CoA, there is always little bit of acetyl-CoA in this motor end plate. Now this is what is generating the end plate potential, like little bit of action potential, even if there is no impulse, even if there is no stimulus that is coming, there is little bit of action potential always that is present in the motor end plate, and it is depolarizing in nature. So this is what is happening always at the junction at the synapse even when there is no electric stimulus. Okay, this is the right microscopic view with this understanding of us. Now, moving on to the electron microscopic picture. Let's understand the electron microscopic picture. Then we will study all the applied portion linked to this. So, an electron microscopic study. Okay, first, what are the what are the parts in a neuromuscular junction? What are the parts in a neuromuscular junction? Just like any synapse or any junction, there are three parts. Okay, there are three parts. First one is the presynaptic membrane. Presynaptic membrane. Then we have, after that, postsynaptic membrane. And between the presynaptic membrane and postsynaptic membrane, what is there? There is synaptic cleft, and it is in the synaptic cleft where we have the miniature end plate potential or motor end plate potential, which is miniature in nature. And remember, this motor end plate potential, it is a local or graded potential, not an action potential. It is not an action potential because it is miniature in nature. It is miniature. It will never cause a contraction of a muscle. Okay, but there will be just small spikes, small current that is present there. So these are the three structures, important structure of a neuromuscular junction. So how does it look like? Okay, so if I have to draw, this is the, it is like an enlarged way, right? This is the axon terminal. Okay, so this is my presynaptic membrane. This is the presynaptic membrane, means this is the axon terminal of the alpha motor neuron. Alpha motor neuron. What does it have? It has got the voltage gated sodium channel. Voltage gated sodium channel, right? What other things it has got? It has got the voltage gated calcium channel. Voltage gated calcium channel. Also, it has the also the voltage gated potassium channel. Okay, these are the channels that is present in the presynaptic region. The sodium channel is little bit further up because the myelin sheath loses after that. Then it has got the voltage gated calcium channel. It has got the voltage gated potassium channel. Okay, it holds it holds synaptic vesicle. Okay, synaptic vesicles. It is holding the synaptic vesicles. Okay. Then we have the gap which we call as the synaptic cleft. Let's keep it little bit wider so that we can draw the other diagrams. Okay. Now on this is the postsynaptic membrane, synaptic cleft, and here this is like the postsynaptic membrane. So these are like if this is the membrane and this is another one. Okay, this is the postsynaptic membrane. Now, what is the unique feature in a postsynaptic membrane? Unique feature in postsynaptic membrane is the presence of receptors for acetylcholine. This is acetylcholine receptor which is NM type. Now this acetylcholine receptor, this is supported by a cluster of protein, So what does this cluster of protein have? What are the structure in that cluster of protein? So the structures are like obviously it has got collagen, it has got the collagen, it has like proteins like agrine, this is agrine, then there is that dock protein for the docking purpose, for the stability purpose. Then we have the musk protein. We also have the rhabsine protein. We have the low density lipoprotein receptor protein. All those proteins are present, which is a cluster, and this is like the collagen protein, which is helping in stabilizing of this acetylcholine receptor. Okay, this is the structure that is present. Now, in the postsynaptic membrane, additionally, what is present? There are this channel, there are these gates for the calcium. So, obviously, first there is this voltage gated sodium channels, those are present, right? Apart from this voltage gated sodium channel, what are present? There are presence of this dihydropyridine receptor, and then there are which are in physical contact with them are the ryanodine receptor. Okay, now both are the calcium receptor. Dihydropyridine receptor is for calcium, ryanodine is also for calcium, but dihydropyridine receptor senses the voltage. This all we will again study tomorrow in details. So these are the structures. So what are the structures we have seen? This is our presynaptic membrane having the synaptic vesicle, having the channels, and the postsynaptic membrane having the receptor for the acetylcholine, having the supporting protein that will help to support the acetylcholine receptor, a cluster of proteins, and then we have the calcium channel, we have the voltage gated channel, and this empty area, this is our synaptic take left. Okay. Now going by the events. What are the events that are happening? What is the event that is happening? So whenever there is like stimulus of threshold level, like electric impulse. Electric impulse means when the action potential wave is flowing down from the cell body through the axon to the terminal region. So when this is happening, there is sodium influx, right? So the sodium influx will cause the calcium entry. So when the calcium enters, what it will do? Immediately it will start the acetylcholine formation with the help of acetylcholine transferase. So how is this formation happening? How is this acetylcholine formed? We will see down. And as soon as the wave of depolarization is over, here through the voltage gated potassium channel, potassium efflux happens. Now, going all the way to understand the acetyl CoA formation. It so happens when there is stimulus to the axon terminal of the presynaptic membrane, then choline plus acetyl coenzyme plus ATP plus glucose. Now, this choline, it comes from the ECF. Acetyl coenzyme comes from the mitochondria in the presynaptic membrane, means of the neuron, and this too comes from the cytoplasm of the neuron. All these things are from the neuron, because we know the neuron has this mitochondria and all these organelles. From there